Okay, so next part is we want to calculate a differential expression for a single cell RNA-seq. You can imagine if you have a single cell data after you do clustering, you can visualize them. You might want to calculate, you say, for example, in this case, this whole big cluster are all CD4 T cells, but you kind of wonder, what is the difference between the bottom half and the top half, right? This is within the same sample. There is no, there's maybe the cell is differentiating, or maybe the drug treatment is making some of the cell move to a different group. The cell identity is still CD4, but there are already definitely significant differences. And so you want to ask, what is that difference? And sometimes you also want to just even ask, different cell types. What is the difference between yellow and orange? That will be a simpler question as well. And so for that, we have to understand the unique properties of single cell RNA-seq. This is from an early review, but I think it captures the result from single cell in general. If you get the readout of the RPM, this is a read per million in that single cell, you can ask, what is the readout, how many, this is by transcript. Every gene, you ask how what's the abundance of, every cell, you ask on every gene how much read count is landing there. Supposedly, these are two SMART-seq created single cell experiments, and they are actually supposed to be identical cells. Even if you try your best, you get a few thousand genes, you look at their expression, you do see some reasonable positive correlation. And yet, you can see it's very noisy. There is high magnitude outliers, some genes that are actually expressed very high, but also very noisy. There's over dispersion. Normally if you think, if this is the identical cell and you do a cell line, you do one replicate, another replicate in a bulk level, you would imagine this curve to be really just on the diagonal. But in a single cell, there's bigger dispersion. And also there are a lot of dots in here that's a 0 in one, but can be pretty high in the other. It's just, in this case, it's high in cell 2, like this spot, but it's not measured at all in the other cell. And so people call this zero inflated Poisson or zero inflated negative binomial from the single cell readout. There are several levels of dropouts. The first is that, there is transcription stochasticity. So if you look at the people who are doing single molecule or single cell experiment, they might notice that some transcript, when they are expressed in a single cell, it has a burst, and then it will rest, and then will rest a little bit, and then a burst, another burst of transcription. Therefore, if you can really measure it on single cell, you might see that this thing lights up a little bit, then will be silent. And that cell will be light up in a different time. And so that's a real biological stochasticity, right? Polymerase getting incorporated and it just finished, and it was wait for the next polymerase to get incorporated to make RNA and so on. So this is, it's biologically, it's real, but then maybe we're not so interested in that. The second one is the technical dropout. In the droplet, as the RNA is attached to the bead in that droplet, and RNA is being labeled, maybe it didn't get a label, or something happened, just didn't make it. And even though in general, if this gene is expressed a lot, is less likely to have zeros, but you can see in these high level ones, there could be noise, and also there could still be dropouts. That's why it's the zero inflated negative binomial distribution. And so for that, there are computational algorithms to deal with this situation. It's called the hurdle model. This can be used for negative binomial or the Poisson. The idea is there is a hurdle. This gene, have to, there's a hurdle that it has to be made into an RNA library. Sometimes it just cannot see it. Then it's zero, it's hopeless. But once it's made into an RNA-seq library, then you try to compare in one group versus the other group. Is there a difference between the expression level of those genes? But there is a significant portion of those that even if the gene is actually expressed, it's still zero. And you can look at the number of cells that this gene has a zero. And also when it is not zero, whether their expression levels are the same or different. And also the propensity, yes, I think this part is kind of the propensity of this being in zero versus being expressed. And so the master algorithm used this hurdle model to calculate. So you have a two-stage, you have to decide for this gene, is it still in the zero part or in the non-zero part? And in the zero part, you calculate the number of zeros, and then the non-zero part, you calculate the real expression level differences. We unfortunately also won't go to any details for this algorithm that is being used. By the way, people are sequencing more and more, and the non-parametric test also kind of works very well. This one is actually very easy to understand. This is called the Mann-Whitney U test, or also known as Wilcoxon rank sum test. So supposedly, we have this big expression matrix. Let's look at one gene at a time. You look at, supposedly, these cells belong to the cluster 1, and these cells belong to the cluster 2. If we look at all of the cells together, we can rank the cells based on the count you read at either TPM or count. You read at that cell, and then you rank all of the cells together, and then you look at the sum of the ranks of all the cells in the one cluster, it's in this treatment or control, and compare with the sum of the ranks of the cells in the other cluster. And the significance is calculated from permutation. But there are some tables you can check. So supposedly you have 10 cells that are normal and 10 cells that are cancer, and they are both in the single cell experiment. Because the 10 cells, and you look at one gene, and the minimum of the sum of rank for the tumor is 55, which means that that 10 cell has this gene expressed as the higher than all the cancer than normal is completely separated. And then 155 is the complete opposite situation. So either is all 10 cancer cells are higher than the 10 normal, or all 10 cancer cells are lower than the 10 normal, right? So the extreme case is 55 and 155. But then you can ask, what is the statistical significance of getting 150? You can then check, there are some tables that they already did the permutation, and they can tell you whether it's significant. These type of non-parametric tests are not so great when you deal with bulk samples, because usually you only have 3 treatment, 3 control. You don't have enough permutations to do this. But with single cell, very often that single cell cluster can have hundreds of cells. Then you can compare one cluster with the other using this permutation. Then it's actually pretty powerful. And you can actually try it on the current homework for Surratt. You can try the mass algorithm. You can also try the Mann-Whitney U test. I would say most of the differential genes you detected are going to be quite similar between the two approaches. Okay, so basically, when you calculate differential expression, if you are talking about within the sample, then you are really comparing different clusters or subclusters, right? Yeah, so it's either within or between clusters. Because one gene across the cell can have many zeros and also potentially many ties, if you want to use a parametric test, which is this zero inflated negative binomial distribution, you can use the masked algorithm. But if you just kind of want to know non-parametric, so non-parametric test does not assume any data distribution. It's less powerful, but since you have so many cells, it can actually work quite well in the single cell setting. When you try to compare across different samples, this is kind of harder. Yeah, so you can, you can still use the within sample differential expression. For example, if you have multiple samples, you can put them all together to run your U map. Then they are all embedded together. And then you could say, okay, let's look at, within this whole cluster, what is the cells in cells in one tissue or one sample versus all the cells in another sample. And then you can still compare their difference. It could be that, yes, they all belong to CD4, but then they are shifted slightly within this bigger cluster, right? So one is slightly expressing something higher than the other. You can still do that. And then so for that, you can still use either mass or man within U test. But you can also get just the expression index from one cluster. Remember, if you really want to know the expression level of some cells, it's not very accurate to do this at a really single cell level. What you can do is you get the cluster and you just merge all the reads in the same cluster together, or the UMI counts. And then you calculate your TPM for the whole cluster. That's actually pretty accurate. This is almost like a bulk. It's called a pseudo-bulk because you merge all the single cell data in the same cluster to form that bulk. And then if there you have enough, like multiple samples, you can get the expression level of this cluster from every sample, and then you can compare your treatment and control in the multiple samples. Then you'll be DE-seq as before, right? Do we need to do multiple hypothesis testing? Actually, every time I ask you, you should be yes, most likely, because we are looking at every gene between the cluster or within the cluster, right? Whether they are differentially expressed. Yeah, so you probably want to remove some cells that are hopeless. They are like so low, always zero, just, yeah. And then hopefully there are sufficiently good number of genes to give you differential expression. And because you have so many single cells, the statistical power could be quite good, even after you correct for multiple hypothesis testing. And then you can do the downstream analysis, such as, after you get the differential genes, you can run gene set enrichment, you can do standard gene ontology that you are doing for homework too. Okay. We're running a lot of little time. So we'll finish the single cell study in the next lecture. And also in next lecture, we will summarize all we have learned about gene expression. And I will show you some scenarios to think about how to analyze data. So you can string those different tools together. Okay.